Hi, I'm Bara, and I would like to invite you all to read our paper in BioEssays. It's called Evolution of Sex Determination in Amniotes, with a question: did stress and sequential hermaphroditism produce environmental sex determination? The link you can find in the video description. We will talk about sex determination and about amniotes. So first, amniotes is a group of vertebrates where belongs mammals, birds, crocodiles, turtles, tuataras, and squamate reptiles. Let's start with something you are surely familiar with: the mammals. In mammals, the sex is determined by the X and Y sex chromosomes, where females possess 2 X chromosomes and males have 1 X and 1 Y chromosome. All eggs bear X chromosomes, and the sex of an embryo is determined by the sperm. The fertilization of an egg by a sperm containing X chromosome leads to the development of a female, a sperm with Y chromosome to a male. In all birds, the heterogametic sex are females. That means that concerning sex determination, all the sperm are the same, but there are 2 categories of eggs. The one with Z chromosome, which is present also in sperms, gives rise to a male embryo, while an egg with female-specific W chromosome leads to a development of female embryo. Well, now it's all going to be a little more complicated. Not only that we can find the 2 previously mentioned systems in other groups of animals, but there is also another mechanism of sex determination, the environmental sex determination, or ESD. In ESD, there are no sex chromosomes, no genetic difference between sexes, and the sex of the offspring depends on some environmental stimuli. The most studied is temperature. The stimuli must act during a sensitive period during embryonic development. That means that any embryo can be, at the beginning, both male or female. For example, in crocodiles, warm temperatures produced mostly males, cold incubation temperatures females. In many turtles, it's mostly just the opposite. Males develop at cooler incubation temperatures, females into warmer. Sometimes ESD leads to a biased sex ratio in a population, and there are concerns that it makes some ESD species vulnerable under the climate change, and that, for instance, sea turtles, there will be a shortage of males. On the other hand, more females than males in a population could be beneficial for population growth. In our team, we are interested in evolutionary history of sex determination in amniotes, and we ask how and why ESD and sex chromosomes evolved. Unfortunately, there is no evidence which we could gain from fossil record, so we have to reconstruct the history mainly from information of inextant species. We know that sex chromosomes evolved many times in amniotes, and in many groups they are dozens of millions years old. There is no transition to ESD in any of these groups, and in fact, there is no well-supported transition from presence of sex chromosomes back to ESD in amniotes, although laboratory experiments documented that some lineages have the potential to do it. From these comparative data, we suggest that ESD could be ancient and could be ancestor for all amniotes. Amniote ESD lineages could have still the same ancestor system of sex determination inherited, of course with modifications, from a generation to a generation for about 300 million years. Well, and if so, there's even more interesting question: what could be ancestral to this ancestor ESD? I would like to draw your attention to a very specific mode of sex determination, the sequential hermaphroditism. Sequential means that the animal can change its sex once in a life. The change usually takes place after a sudden change in social environment, mainly after the death of the dominant individual in the group. We pointed that there are intriguing similarities between ESD and sequential hermaphroditism, like absence of genetic difference between the sexes and often biased sex ratio in a population. We hypothesized that ESD species and sequential hermaphrodites could also share the molecular mechanism of sex determination linked to stress. In ESD species, we often incubate eggs at stable temperatures, which is under many conditions unlikely in nature. Certain extreme constant temperatures could be seen as a stressor. And in sequential hermaphrodites, it has been already shown that the sex change is linked with stress and stress hormones. Could be the common ancestor of Amniotes sequential hermaphrodite who started to change sex already during embryogenesis according to the level of stress in the environment? Well, it's a hypothesis supported up to now by some indirect evidence, and if you are interested in more details, go and read our paper.